So what we would like to cover over the next hour or so is some of the ongoing work into understanding neural development. And the focus is going to be on the spinal cord and cells that are involved in locomotor control. And I'm just going to point out a few of the high points by way of introduction for myself and the other speakers that are going to soon follow here. We're really going to talk a lot about the developmental biology of stem cells, less from the view of regenerative medicine and replacing cell types, but more from the view of trying to understand molecular pathways involved in stem cell differentiation. And the logic behind this is that we hope that this will lead to a better understanding of normal developmental processes in human embryonic development, that this can lead to optimized cell differentiation in vitro for regenerative strategies, that this can lead to the identification of novel target pathways that might be manipulated, and also that we can use this system as a way to model diseases to gain novel insight into the disease pathways as well. Most of the experiments you're going to hear about are going to be in the context of mouse embryonic stem cells differentiating into neurons. Why mouse? Because this is a particularly powerful genetic system. The other feature that you may or may not be aware of is that obviously the embryonic development of a mouse is much more rapid than a human, and a lot of these features are replicated in vitro, and therefore differentiating mouse embryonic stem cells in vitro is a much more rapid process, which means that experimentation goes a lot faster. One of the overarching questions that you'll see handled during this session is really a question of neuronal diversity. So there are on the order of 100 billion neurons in the brain and spinal cord, 100 trillion connections. These are obviously enormous numbers in the context of 25,000 genes. So the bioinformaticists out there would say 25,000 genes, that's a lot to think about. But in the context of the number of cells and connections in the brain, that's actually a pretty small number. And this leads to the question of how all of the complexity in the nervous system is created with what amounts to a relatively small number of genes. And the speakers are going to be touching on some of the pathways that control neuronal differentiation. The system that we're going to be focusing on is, as I alluded to, the spinal cord, and particularly neurons that are involved in motor control. So the spinal cord is comprised of a wide variety of cell types that can be characterized by many different features. The final output that is controlling our movements are spinal motor neurons, whose cell bodies reside within the spinal cord. They grow axons out to the periphery where they form synaptic connections with muscles. And it's really the disruption of the activity, either the inputs onto these cells or directly the survival of these cells, that lead to a variety of things like motor diseases, but also in the context of spinal cord injury, where you disrupt the input onto these cells. What do we know about the normal embryonic development of the spinal cord? Well, over the last 20 years, there's been enormous progress in understanding some of the cell-cell interactions and intrinsic genetic programs that control the differentiation of the spinal cord, which is why it is now such a powerful model system for studies with stem cells. One of the observations has been that the notochord, a structure right underneath the developing neural tube, is a source of inductive factors that control spinal cord development, and the identification of that inductive factor, or the inductive factor involved, is sonic Hedgehog. And sonic Hedgehog is a secreted molecule that acts in a graded way to control the differentiation of very specific cell types in the ventral neural tube. And as these concentrations of sonic Hedgehog are being read out, it activates different transcriptional programs that are really controlling cell identity. So here are the speakers that will be talking over the next hour or so. Henk Roelink is going to cover some of the signaling interactions that drive this process of stem cell differentiation. Todd McFarlane is going to talk about some of the intrinsic transcriptional programs that are involved in controlling cell fate. Ben Novich is going to touch on how you generate different subtypes of neurons, and in particular motor neurons, because the cells, motor neurons that innervate different muscle targets actually have different qualities from one another. And I'm going to end the session by talking about some recent work on regulatory pathways that are involved in maintaining motor neurons and their survival. So I'm going to turn it over now to Henk. Here's a pointer.
Thanks, Sam, and briefly for the invitation here. All of you see, this is going to be basic science. So, as Sam mentioned, we're interested in sort of how this hedgehog, where the RNA is shown here in the notochord and the floor plate, makes a protein that establishes in a gradient, and then this gradient is interpreted in the stereotypic differentiation of cell types in the ventral spinal cord. For those who don't know, this hedgehog gene got its name because the leading mutation in flies, where you have a fly larvae here, if you miss hedgehog, the larva looks like this, with these little denticles everywhere that is reminiscent of this European or insectivore that is very common in Europe that has these tentacles everywhere. The other thing I should bring out early is that this is a mouse where you can see the expression pattern of hedgehog during development. We're going to talk about this expression there in the floor plate and there in the notochord, but realize it expressed at many sites. And hedgehog expression in developing hair follicles, here the whiskers is shown. And I should also say that almost at all these sites where hedgehog is expressed during development, later on hedgehog contributes to tumors in these tissues. So all the signaling interactions I will talk about are not only important in development, but also in the understanding of how these tumors arise. So here's the hedgehog protein in the notochord, in the floor plate, and you can see that it distributes nicely away from its sources of synthesis. The idea is it establishes a gradient, and this gradient is interpreted here in the cross-section of the neural tube with cells that are very close or are the sources of hedgehog responding to a very high concentration, and lower concentration gives you the specific cell types, and especially this group is going to be of great interest, is labeled here with this transcription factor HB9, which are going to be the motor neurons that are going to connect to the muscles. Now, the hedgehog response pathway is a little bit counterintuitive. It's sort of a double negative. So the idea is the following. The ligand binding domains are patch 1 and patch 2, and in their unliganded state, they inhibit a GPCR, a G protein coupled receptors called smoothened, and keeps it in an inactive state. So in a diagram, you have that here. The idea is that binding of hedgehog to patched binds hedgehog very well. There's an internalization of this complex, smoothened, travels through the cell, might make it to this primary cilium, and organize a transcriptional response that is mediated via a set of zinc finger binding transcription factors. And so the loss of patch 1 and patch 2, especially patch 1, in a variety of systems has shown that leads to the activation of smoothened. Now, downstream of smoothened are a number of things, like these motor neuron markers, but patch 1 itself is downstream of this response as well, and it becomes a little bit later on in the talk. Now, obviously, this interaction between patched and smoothened is really important in development and in cancer, and again, it's a bit strange. It turns out that patched is actually a member of a highly conserved group of proteins, which are the so-called R&D transporters, which are proton-driven antiporters. They're present in every cell in the world. Now, what all these cells have in common is that they use a pH gradient across a membrane to efflux a small molecule to the outside world. And here, in bacteria that are gram-negative, to cross the second membrane, there's actually a chimney on this molecule, but it's all the same. They have 12 transmembrane domains, and the molecules function as trimers, so this whole complex is huge, and they swap a proton from the outside and then toss a cargo through the central channel to the outside world. Now, these are proton-driven antiporters, and again, every antiporter in the world, with one exception here, has a very characteristic aspartic acid in the 4th transmembrane domain, which is involved in binding of the proton on its way from the outside the cell to the inside the cell, and this proton is exchanged for its cargo. This is important because mutation of this aspartic acid uniformly leads to the killing of this antiproton antiporter activity. Now, patched also binds Hedgehog, and so to look at these various functions of patched, these are 4 mutants that we using in the rest of this little talk. There's a form of patched that misses the Hedgehog binding domain. We call this patch 1 delta loop 2. The antiporter function is intact, so this cannot bind patched, but it still can pump whatever it pumps. Then we have this mutant where the antiporter function is killed, but a Hedgehog binding loop is intact, and one where these 2 mutations are combined. Now, if we put these cells in the neural tube of a chicken embryo, and we do that very simply, we inject the DNA in the lumen, we expose these embryos to an electric field, the DNA moves towards the positive electrode, the electric field makes little holes in the cell membranes, and DNA is internalized and gets expressed. And so it's a very rapid and easy transgenic model. If you misexpress patched in this way, you can see that these red cells, which actually PAX7 cells, and indicate very low or absence of Hedgehog response, are slightly expanded, which sort of makes sense because patched is a negative regulator of the pathway. When we take this Hedgehog binding loop 2 off, we get an incredibly strong molecule, patch delta loop 2, and you can see wherever this molecule is expressed in green, PAX7 is on as well. In other words, if you express patch 1 delta loop 2, you do not respond to sonic Hedgehog. And this is 100% dependent on this antiporter activity, because if we kill the antiporter in this mutant, and you get this double mutant, it is dead. The antiporter activity is probably also responsible for this minor inhibition, because if we just mutate this antiporter in the wild-type patched, we have yet another dead molecule. But important of this experiment is that the inhibition of smoothened requires the antiporter activity of patched. Now, the problem with these experiments is they are done in an environment that contains endogenous patch molecules, and I showed you they trimerize, so we don't actually know how these interactions take place. So realizing we need to study this in an environment that is null for a variety of these molecules. Sam showed there's a very straightforward procedure where a mouse ES colony is trypsinized, reaggregated in a completely defined medium, and a little bit of RA makes these balls of cells that very closely resemble an early developing tissue in the spinal cord. And here's a notochord, it makes Hedgehog. A couple of these embroid bodies are around it. This is a live image, and you see that GFP is induced very nicely, and this GFP is actually expressed exclusively in motor neurons, and here's after staining for Hedgehog. So a very simple mechanism that allows you to study these signaling interactions in vitro. Using these new genome editing tools, we made a large collection of embryonic stem cells that were either lacking patch 1, patch 2, Hedgehog, dispatched. Dispatch is, don't worry much about it, but it is required for the secretion of sonic, Indian, and desert Hedgehog, and which are null for smoothened as well. Now, these are, of course, cell lines you could never make a mouse of. They are completely unusable as such, but they are very useful in vitro. We made these cell lines by reiterative use of TALENs and CRISPRs. The way we do it, we transfect it, we transiently select, we pick up colonies, look at the genotype, and turn this crank as many times as necessary to knock all the genes out we want, and our record is now cell lines that are null for about 6 of the genes in the early Hedgehog pathway. If you look at the differentiation of these cells, they actually support the model quite well. In a sense, here's wild-type cells. They acquire a relatively non-ventral phenotype, as indicated here by these green cells, PAX7, which define dorsal cells. If we remove one allele of patched, we're getting ventral cells here indicated by these red and blue cells, and suppression of these green cells, so no dorsal cells. If we remove smoothened, the required GPCR, these cells revert into a dorsal phenotype, and if we remove patch 2 on top of that, we actually lose a certain Hedgehog sensitivity that is still present in the patch 1, patch 2, in the patch 1 null cells. But they very much support the idea that patch 1 and patch 2 are awesome inhibitors of smoothened, and the loss of these inhibitors activates the Hedgehog response pathway. Now let me reiterate, these patch molecules are proton-driven antiporters, and what they do is they take a proton, they let it into the cell, and they flux the cargo out. I showed you that this proton-driven antiporter is required for the inhibition of smoothened, and so the question we were really asking is, can patched secrete a cargo that inhibit smoothing in adjacent cells, right? Is the function of patch what we call non-cell autonomous? And the way we do that is that we're making these combined tissues where we mix cells with distinct genotypes into an embryoid body. And what we're mixing here are cells that have an upregulated Hedgehog response pathway due to the absence of patch 1 and patch 2, and surrounding them with cells in which we vary the amount of patched alleles that are present. Alternatively, we can use cells that respond to Hedgehog and include Hedgehog-expressing cells. And again, these gray cells, we can do whatever we want with them. But the essential thing is we measure the Hedgehog response in these cells, and we change the patched levels in the cells that are surrounding it, right? Now, the readout cell that we're using here is a patched 1, 2, Hedgehog triple null line. And I should say that I told you that patch 1 itself is upregulated in response to Hedgehog signaling. And this particular allele of patched 1, which is a null, but it drives leg Z of the patch 1 promoter. And so it can be used as a measure of the level of activation of the Hedgehog response. If we turn these cells into neural tissue, after about 2 days, there's a lovely induction of leg Z, and it sticks around for a while. Makes perfect sense because the inhibitors of smoothing are absent. Now it gets interesting. If we take cells and we make an EB that contains about 10% of these patch 1, patch 2 null cells and surround them with cells that are always dispatched and Hedgehog null, so there is no signaling that is mediated by Hedgehog or any of the other ligands, but either have both patch 1 and patch 2, only patch 2, or neither patch 1 and patch 2 in the surrounding cells. When patched is absent from the surrounding cells, we get a lovely induction of leg Z, but when patched is present in the surrounding cells, the induction is a lot lower. This still works at a 50-50 distribution of cells where we read out the response and cells where we change patched contribution. Even at 10% of the cells that contain patched are able to somewhat suppress the Hedgehog response in the surrounding cells. Now, this is really crazy, right? We're having now a primary receptor for Hedgehog, the ligand-binding molecule, that is actually able to inhibit the Hedgehog response in adjacent cells, right? These cells have no patched, and so the surrounding cells do, and they listen to patch in the surrounding cell, which is pretty much consistent with that patched actually effluxes an inhibitor of smoothing that works on adjacent cells. This even works when we use the readout cells as wild-type cells. So here again are cells that express a motor neuron marker that is, or a motor neuron-specific promoter that's driving GFP. If we take these cells, 5%, 95% are patch 1, patch 2 competent. We find very little induction. If they miss patch 1, still very little is happening. But if we take this situation and we're adding a small molecule activator of smoothing, it's called SAG for smoothing agonist, we can see that the difference of the response in these cells to the smoothing agonist is a lot stronger when the surrounding cells don't have patch 1 and patch 2. Right, so we're in a situation that the patched status of the surrounding cells determines the sensitivity of wild-type cells to the smoothing agonist. And the same is observed if we include a small fraction of Hedgehog-expressing cells, which themselves are patch 1, patch 2 null, in the environment of these cells, and where the gray cells have patched, there's very little induction. The gray cells have no patch, there's a strong induction, which means that the patch status of the intermediate cell strongly affect the ability of the blue cells to respond to Hedgehog released by the red cells. Now, we can actually block that by the inclusion of a small amount of patch-containing cells in the surrounding cells. Which brings down the old model of Hedgehog signaling to one that looks much more like this. We're having patched, which can inhibit in trans, in adjacent cells, this critical molecule smoothing. The cis inhibition exists, but it's not that important. And it looks like that upon binding of Hedgehog to patched, this inhibition is gone, which results in smoothing that is now way more sensitive to respond to sonic Hedgehog in itself. Now realize that patch binds Hedgehog, and there's a wide literature about patched acting cell autonomously, meaning that patched acts in the cell that is responding to Hedgehog, right? So what I described just is that patched actually acts in the neighboring cell to affect the Hedgehog response. And so we wanted to know the cell autonomy of patched, how these mutants were involved in there. And we did it the following way. We took our patch 1, patch 2 null ES cells. We derived some fibroblasts from them, which is a fairly straightforward procedure. And this time, we transfected the described patched mutants in with a glyluciferase. So now the cells that express patched, or these mutants of patched, they are the only cells that we assay for the Hedgehog response. So we purely look at the cell autonomous activation of the Hedgehog response in the transfected cells. Realize all these cells are patch 1, patch 2 null. So we do 2 experimental conditions. One's where there are no Hedgehog-producing cells included in this experiment, and the other one where Hedgehog-producing cells are included in this experiment. And these are just patch 1, 2 null cells that are separately transfected with Hedgehog, mixed in with the culture. And now we assess the ability of patched to mediate the cell autonomous response to Hedgehog that is provided in nearby cells. And it might not be a big surprise that if we put patched wild type back into these cells, they respond to Hedgehog quite nicely, and that is all good and well. However, this form of Hedgehog that is competent for the antiporter activity but cannot bind Hedgehog, so has the ability to inhibit smoothend, is completely blind to Hedgehog that is supplied from somewhere else. In contrast, a form of patched that cannot inhibit smoothend via the pump activity because it is mutant for this critical aspartic acid, restores Hedgehog signaling just fine, and a double mutant cannot. So whereas we first found that these non-cell autonomous inhibition of smoothend required this R&D pump activity, we're finding that the cell autonomous restoration of Hedgehog signaling to cells only requires the Hedgehog binding domain, but does not require the pump. So to look a little bit more at that interaction between the patched loop and Hedgehog itself, we tested 2 Hedgehog mutants. One is E90A, which cannot bind to a set of co-receptors that is also involved in the internalization of Hedgehog. And this Hedgehog H183A, this form of Hedgehog cannot bind to patched. And if we test the activity of these molecules, you very nicely see wild-type Hedgehog induces a lovely response. If you take these mutants, the cells don't respond to it because they can't bind it. However, if we take these Hedgehog mutants and we transfect them straight into the patch 1, 2 null cells, so now we're having the ligand available in the same cell that we are assessing the response in, and these cells have no patch 1 and patch 2, we're getting a very strong induction. Also by the mutants, showing that the interaction with patched and Bock and Kiddo are of very little influence there. Right, so now it looks like, and our model is that for the cell autonomous response to Hedgehog, the pump is not that important, but Hedgehog is internalized by patch 1 and patch 2 to a compartment where Hedgehog is handed over to smoothend where it can be activated. And it is this internalization event that we can circumvent by direct transfection of Hedgehog into those cells, which now doesn't care whether it can bind to patched or to Bock or Kiddo to nevertheless activate the Hedgehog response very strongly. Now, the next slide, the pictures are not as nice, and I can tell you exactly why this is. But in vivo, the same things happens. So here we have a wild-type Hedgehog that is transfected into the neural tube, and you see this massive expansion of these red cells, which actually NKX 2.2, mean that this whole side of the neural tube responds as if it sees high levels of Hedgehog, which makes sense. This Hedgehog mutant, which cannot signal when you add it to cells, does exactly the same thing. And so does this one, does exactly the same thing. So it looks like that these Hedgehog mutants, when they are supplied inside the cells proper, in the spinal cord, they induce the Hedgehog response just fine. Now this section is really cruddy because the student that did this, Astrid Alfaro, came steaming into my office and said, I spent months characterizing these mutants, and they were great in vivo, but I put them in, or in vitro, but I put them in vivo, and they just work. And I said, wait, we'll find the answer to this. And the interpretation of this experiment right now, that this Hedgehog, which cannot bind patched, nevertheless induced the Hedgehog response by circumventing this whole machinery, and interacting either directly or not quite directly to smoothend. And so whereas the old canonical model is not wrong, we think it is highly incomplete, where Hedgehog binding to patched releases a direct inhibition of smoothend. We think that there's 2 steps in this phenomena right now. The first step is that there is a non-cell autonomous inhibition via the proton antiporter activity in which patched inhibits smoothend. This inhibition is released and which then results into a prime state of smoothend. Now, independent of the antiporter activity, Hedgehog binds to patched and is internalized, which then results in the presentation of Hedgehog to smoothend in a way that we don't know yet, which results in the activation of the pathway. So the lesson here is, and where we are right now in this thing, is that we're getting this beautiful pattern of these discrete cell types that are induced by this gradient of Hedgehog, but the complications right now, which was figured out with all these cell lines, is that we have a Hedgehog gradient in a neural tube, which results in a patched 1-2 gradient, which itself causes an inverse sensitivity gradient to sonic Hedgehog, which is then interpreted by these cells again in a patched 1 independent, in a patched 1 dependent manner, but the activation is patched 1 independent. And all these effects together, nevertheless, result uniformly into this beautiful differentiation pattern in the neural tube. I have a couple of sort of larger take-home messages. The first one is mind the paralogs. Each gene in the genome has genes that, almost each gene in the genome has genes that look alike it, and it made a world of difference in the interpretation of our experiments to not only remove patched 1, which in a mouse gives a great phenotype, but also remove patched 2. And the second one is that cells talk to each other all the time and do that in possibly rather unexpected ways. We had not predicted that a fundamental regulation of the Hedgehog pathway was actually the effluxing of a small molecule by what was thought to be a relatively standard receptor for the Hedgehog response. And I'll stop with the people that did the work, 3 great undergrads, grad students, Esther Alfaro, currently at Genentech. She analyzed the mutant cell lines. Brock Roberts did an heroic effort as a CERM fellow to knock out all these proximal genes in the Hedgehog pathway, and Catalina Casillas, who did the analysis of the patch 1, patch 2 null cells and showed that they still respond to Hedgehog. We got on track of this by receiving cell lines initially from Matt Scott and Andy McMahon, which showed that the genetic modification of these ES cells in combination with this in vitro differentiation is actually a very fertile pathway of research. And with that, I thank you.
So there are, for those of you at the back or on the sides, there's plenty of seats here at the front.
Roger, the way we're going to try and do this is allow the speakers to take 2-3 questions that are very relevant to what you just heard, and then we'll also allow you to ask more general questions of the entire group toward the end, based on.
That's as we had with Larry's thing, but we ran out of time for the panel at the end. So if there are any questions, there's obviously one here.
So do you think this is important for those of us who have been using sonic Hedgehog versus smoothend agonist to differentiate cells? Because it sounds like these two, they might have very different downstream signaling. If you're using Hedgehog, which can work via patched, and obviously the smoothend agonists aren't touching patched at all.
I think it is. But the idea with the smoothend agonist is that it binds to the same site on smoothend as where the cargo of patch binds. So patched and the patched cargo and SAG are actually in a competition for that binding to smoothend. So how cells respond to SAG is highly dependent on the patched environment that those cells are in. So it does make a difference.
Yes, in terms of the last part of your speech, you talked about the cell-to-cell interaction when you graft a embryonic stem cell into a particular area. Now, have you actually isolated which microRNAs, which exomes, which cytokines are necessary to induce the pathway in which you're looking for properties of regeneration?
I'm going to defer that question because those are going to be partially addressed.
Okay, thank you.
Yeah.